Well, first of all, I'm very happy to be the host for this episode of Future Frogmen Conversations. And as Richard said, today's conversation is going to be with Katie Martin. Actually, Katie and I have actually known each other for a very long time. 2011, I think, right? Katie was a sophomore in my Gen Bio 3 class way back when, 8 years ago. And over the course of 8 years, Katie and I have had a lot of adventures together. She's basically taken, I think, every single class that she could have possibly taken with me. We've had a lot of adventures, both in the classroom and especially in the field. So I'm very excited to hear about what Katie's doing now. But first of all, what I think we should do is maybe hear from Katie. So Katie, why don't you tell us a little bit about yourself, and not just about yourself, but also your academic background as well.
Yeah, so I mean, I grew up, I was one of those kids that was crazy about animals and crazy about nature. And, always really loved my science classes. And, throughout high school, I kind of grappled with, what am I going to do with this inclination and with science later. I think that a lot of students that are good at science or good at math are kind of told like, oh, well, maybe you should consider becoming a medical doctor, or, it's kind of you're like fast tracked in that direction. And, entering Fairfield in fall of 2010, which feels like forever ago at this point, I was kind of, weighing my options. I was like, okay, keeping the idea of a med school path in the back of my mind, but at the same time, my love for nature and, viewing biology as a puzzle that I really liked solving never went away. And so it was during my sophomore year that I took Gen Bio 3, and that's where I met you, Ashley. And we got to talking about how you bring undergraduates to the field, specifically to Brazil, about every other year. And so for me, that was kind of like a really important crystallizing moment for me of, okay, well, let's give this pathway a shot. And I ended up going to Brazil for the first time in January of 2013 as part of your tropical zoology field course. And the rest is kind of history, as they say. I graduated Fairfield in 2014, and I took a gap year. I worked for a little bit just to take a breath of air and just to kind of figure out what my next steps are going to be. I began my master's in the fall of 2015 and completed it in the spring of 2017 at Stony Brook University in Long Island on New York, and took another gap year while I applied to PhD programs. And then in the fall of 2018, I started here at the University of Central Florida in the Department of Biology.
Awesome. So yeah, so you're currently working with Doctors Kate Manfield and Anna Savage, right?
Yes.
Why don't you tell us what you're working on right now?
Sure. So I'm broadly interested in evolutionary biology, and I consider myself an evolutionary biologist. Right now, I happen to be working on sea turtles, and what I'm really interested in is how sea turtle immune systems have evolved. And one, I'm interested in that because we don't know a whole lot about reptilian immune systems in general. So that's kind of your more like basic science, why should we care approach. But then on top of that, there's the applied reason of the fact that sea turtles, like a lot of wildlife in this modern era, are afflicted by this emerging infectious disease called fibropapillomatosis, which is just a big word for these really gnarly tumors that grow on the sea turtles, both inside and outside of their bodies. So that's another more applied reason to kind of care about their immune systems and see what's going on in the context of diseases they face.
Can you tell us a little bit more about these fibropapillomas?
Yeah, I actually have a couple photos that I can share here. And they are pretty gnarly. They're kind of like these giant cauliflower looking things. Mm-hmm. So you can kind of see here growing on the soft surfaces of the sea turtle, and even on the cornea, on the eyes, are these tumors. They can grow pretty large. They can also be small. What's really interesting about fibropapillomatosis, or FP, is that it can recede. So we might encounter a juvenile turtle, say in 2014, and it's got these paps these tumors growing. And then we might encounter them again in 2017, and we see evidence of the paps having completely receded. So there's this really interesting dynamic of, you know, is it kind of like human chickenpox where, you know, you get it and you clear it and then, you know, you're okay for the rest of your life? What's going on with this? And then here's another photo of a turtle that's actually in rehab. And you can see some of these huge masses, again, growing all over the soft tissues of the turtle. So these tumors like to grow on epithelial surfaces, so things like the skin, but they'll also grow internally, meaning that they can grow on the tissues of the digestive system. And so while we don't say that this tumor disease is malignant per se, it can absolutely cause sublethal effects. So it might slow the turtle down. It might impede their ability to feed. They might not be able to see. So it absolutely can lead to morbidity.
Does this affect all species of sea turtles, or in particular a specific species?
So it has been found in all species of sea turtles, but the thing that's interesting about it is that it most impacts juvenile green sea turtles, the green sea turtle Coloniomytis. So while it has been noted in all 7 species, it's really just that species and at that life stage as a juvenile. Yeah.
Interesting. So why do you think that it's affecting just that life stage?
Yeah, so that's a really good question, and one that we're trying to work to disentangle, and one that hopefully my dissertation will contribute to. So when we're talking about a disease, we're always talking about it in the context of what we call the disease triangle. So that is the interaction of the host, the pathogen, and the environment in which that interaction occurs. So it's very possible that this has to do with immunogenetics, that this has to do with the type of immune genes that a sea turtle has in its repertoire to fight off pathogens. It also might be that this is just something that all juvenile turtles, especially greens, encounter. Kind of going back to that analogy with chickenpox, it's just something that if you're a juvenile sea turtle, you're a juvenile green sea turtle specifically, you're just going to get it in the same way that human children just tend to get chickenpox. There's also this interesting interaction with environment. So when a sea turtle is born, it climbs up out of its nest, and for the next 5 to 10 years after it hits the ocean, it just hangs around in offshore habitat. And we refer to these years in sea turtle biology as the lost years, because we're not really sure what these little tiny turtles that are about the size of a dinner plate are doing. Some satellite tagging has more recently shed light on what's going on and how they use their habitat, but for the most part, they hang out in these giant sargassum seaweed mats in the Atlantic, and that's where they have protection, and that's where they eat. But when they grow a little bit larger and enter their juvenile stage, they begin this transition from offshore to nearshore. So they come to nearshore waters like estuaries and lagoons and really shallow neritic zones that are warm and that also have a lot of food. However, that is also the area where there tends to be a lot of interaction with humans. So are they getting this disease simply because they're a juvenile? Or are they getting this disease because when they come into these shallow areas, this is the first time that they're encountering pollutants and other stressors mediated by human presence, and that's just like, you know, the last straw for them, and they're immunocompromised and develop this disease. So that's still very much a question that we're trying to figure out.
Right. So I know that this particular disease is pandemic, from what I understand. So it's found everywhere. But are there specific places or locations where the incidence of FP is very high?
FP has been on the rise on the Atlantic coast of Florida since about the 80s. And the research project that I'm a part of has been also monitoring in the area since about 1982. That's the Marine Turtle Research Group at UCF. And what we have seen is that in the particular system that we study, the Indian River Lagoon in Florida, which I can show you a couple pictures of in a second. It's right here with this star. This is the Indian River Lagoon, and we see that the incidence of FP in turtles, in green sea turtles in the lagoon, is about 50%. It's a bit lower, I think around 20 or 30% for loggerheads, but I'm not sure entirely if there's different hotspots. I know that in Hawaii, the sea turtles that bask on the shores there, they have a pretty decently high incidence of FP. So I think there is a geographical structure to this, but at the same time, it might have less to do with where on the globe these sea turtles are, but rather what environments they're currently inhabiting. So if you're in a coastal and warm area, maybe it's just kind of a given that you're going to develop FP.
Can you explain to us what actually causes FP?
Yeah, so this is a really interesting disease. I mean, as I mentioned, the tumors can recede and, grow and then recede. And we know that in part it's caused by a herpes virus. So this is the same family of viruses that causes certain cancers in humans and even cold sores in humans. So it's caused in part at least by that herpes virus. And we've shown that, anytime that the PAPs are present, so is the herpes virus. But we've also seen cases where there are turtles that might be virulent, meaning they have the virus, but they don't show any symptoms. So this really interesting modulation between being asymptomatic and symptomatic. We also think it's possible that there are these tiny little marine leeches. I mean, they're really nondescript, tiny, thin, and white, that might be mechanical vectors of this virus, meaning that they might go from one turtle to another, and by feeding off the blood on the turtle, they are spreading and introducing this virus to other turtles.
Oh, really? So
yeah.
this particular leech, is there a specific species of leech that does?
Yeah, what's really interesting is that they're in the genus Osobranchus, and we have an undergraduate in the Savage Lab who is actually looking at species that might persist on green sea turtles versus loggerhead sea turtles, and that perhaps, there's some kind of correlation in leech species. So I think she's been analyzing not only the viral content of each leech, but also what types of turtle it's found on. And she's prepping that manuscript right now, but I do believe what they have found is that these leeches are pretty globally distributed, but there does seem to be a high fidelity in species. So this one particular Osobranchus leech species nearly is always found on a green sea turtle. There's not much crossing of the leeches and on their hosts.
Oh, interesting. And I don't know how much work has been done on these leeches, but do you find that there's a higher prevalence of these leeches in these areas where the juvenile sea turtles are actually found?
That's a good question. I'm not sure that it's been really investigated in the literature, but I will say, if you were to see these things, they're like super tiny. I really can't imagine them existing on their own in the water column, although I'm not a leech expert. So I would imagine that if there was a time and a place for leeches to cross, say from one green sea turtle individual to another, it might be when these turtles are in slightly closer proximity than they would be otherwise, which would be in shallow areas where they've all come to aggregate and feed. So it's possible that that these leeches just do really well in these shallow, warmer areas. Yes.
You have evidence that some individuals can actually be asymptomatic even though they have the virus. I'm assuming that once they get it, they never really get rid of that particular virus. Is that right? Like it just sort of hides out in their system. Is that correct?
That's a good question, and I'm not sure entirely. So the thing that's interesting about this too is that, we, especially as part of the Marine Turtle Research Group, we regularly encounter adult green sea turtles and adult loggerhead sea turtles. They're females that are nesting. We almost never see the fibropapillomatosis on their bodies. There's almost never tumors on the adults that have nested. Now, is that because they've cleared the infection, or is it because individuals that had the tumors didn't survive to reproductive age? That's an open question. We certainly have the blood samples where we could, from these mothers, where we could test to see if the virus was circulating. But honestly, off the top of my head, I am not sure that anyone has actually gone in and confirmed that, at least for our population.
It appears as if these turtles are actually getting this through this leech, this mechanical vector. But once they actually get the virus, if they don't get rid of the virus, they're sort of asymptomatic and you're a mother, is there a possibility of there being vertical transmission as well with this virus?
We think that there might be, and actually a project that we just got permits for this past summer and just began this past summer was to basically, so as part of our monitoring project in Brevard County, along the coast in Melbourne Beach, we monitor how many nesting mothers come up on shore. So we not only count the number of tracks that we see in the morning, but we also have a certain number of mothers of each species that we have to track and mark every year. So by marking, what we do is we wait for a sea turtle to come up on shore, and we work her up, meaning that once she's started to lay her eggs and she's in this kind of trance that turtles get in when they're mid-laying eggs, we take skin, we take body measurements, all that sort of thing. But in addition, once she leaves, we also then follow up with her nest, meaning that we wait from the moment the eggs were laid. We wait about 60 days after they've been laid, and we'll go and assess the success of that nest. Mm-hmm. So the project that we recently started actually involved taking blood from the mother, and then also taking blood from one of the hatchlings, which is very tricky. And that's part of the reason why the permitting process took a little longer, because, I mean, you have a hatchling this size, you need to be really careful that you're not exsanguinating the hatchling. And so we had members of our lab trained and permitted to do this work and take a little bit of blood from that hatchling and to try to test to see, if mom has the virus, do the hatchlings as well. Because it is possible that this is a virus that maybe a hatchling can be born having.
Right. Okay. So I know that this is kind of a sort of aside from the actual work that you're doing.
Yeah.
So the actual work that you're doing now in relation to FP, do you want to describe to us what you do?
Yeah, sure. And for that,
Thank you for watching the Future Frogmen Conversation Series. Please check out our website at www.futurefrogmen.org.